dark? Well, let me know. I don't know if we need to dim the lights. Can you hear me? That's good. I'll try to speak up a little. More like wearing a microphone. Tethered to that. So I'm going to talk today a little bit about my research, and as I was discussing earlier, Dr. Betts, is that I'm really living in the world between chemistry and biology. So hopefully there's something for everybody in there. I'm in a chemistry department, but as you'll see, what I do is use the biology to kind of probe that chemistry and find molecules that are interesting. And that's the part of my research that I want to talk about today. And that's what this sort of long, admittedly long title is about, trying to fish out some of that chemistry. So what I'm going to do first is to talk a bit about this general area that I'm interested in. These are so-called harmful algal blooms, specifically some things called cyanobacteria, or blue-green algae, and try to impress upon you that they're very important for human and environmental health. And then talk a bit about how we've used against this biology, specifically something called the zebrafish embryo model, to really delve into that chemistry to do a lot of new molecule discovery, which is part of what we do, a big part of what we do in my laboratory. Okay, first I want to start off with I'm going to talk about what we do, and I would say it's the big we in quotation marks, because a lot of this work is done by students, graduate students and undergraduates. I got to say, they don't even let me in the lab anymore. Save to water plant that's there. Because they're really the ones that are doing all of the work. So all of this we is going to be a lot of people, and this is the most recent picture I took just on Friday, actually, for something else. And some of the faces here. So the work that I'll talk about is specifically going to be some work of Asha, Jaja, Katie who's not on here. I think Chris has been involved in some of this work. And I'll have a chance to talk about other projects, but certainly happy to talk about it afterwards. And this is the most recent picture of the lab. The reality is there's been a lot of people. I like to put up here this list of undergraduate students that have involved in this. We've had a number of them over the year who have contributed to this work in one way or the other. Gone on and not the photo now, by and large. I'll see a couple of them. So I also really want to acknowledge them. And it'll also kind of pursue what I'll talk about later, and that's research opportunities at FIU if you're interested to come to that small session after the lecture. And of course, I want to acknowledge some of our other collaborators, both locally at the University of Miami, nationally, some at Baylor who have been involved in this work, also some folks doing some research at NASA's JPL. We'll talk about that work today. And internationally, some work we've done in Mexico. Just so you can see their names. And of course, the people who pay the bills. We've been funded by the NIH, NOAA, and USDA over the years for this and this research and some other areas of research. So as I mentioned, I'm going to talk about algae. And I want to explain a little bit what they are, in case you don't know what algae are, because they're sort of a strange group in that they're not a distinct taxonomic grouping. So really, what are these algae? This is the sort of five-kingdom view of the world, is the way I learned it. And the algae really fall between two of these kingdoms. The protists, the single-celled protist organisms, and the prokaryotic monera, or the bacteria. So they actually span two whole kingdoms. That's why I say they're not a really short, defined taxonomic grouping. And within this, we also get these other sort of operational classifications like the macroalgae. And these are larger things that are in the protists that have plant-like structures. None of these are plants, but all the algae are tied together by the fact that they're photosynthetic organisms, and truly they're actually single-celled photosynthetic organisms, but some of them actually have plant-like shape to them, and it's probably what you know as a seaweed, or we call them the macroalgae. Operationally, though, we also parse out this group called the microalgae, and these are the things that are truly single-celled photosynthetic organisms. And although they're small, you'll see that they can form large, massive blooms. And specifically, this includes a number of these protist groups, but I'm really going to talk mostly today about these prokaryotic cyanobacteria, which are near and dear to my heart, and the primary thrust of what we do in my lab. Okay. Now, I mentioned this bloom concept. I will talk about what non-cyanobacteria thing. That's just to give you the idea of what these algal blooms are all about, these harmful algal blooms. And for those of you who live in Florida and grew up here, maybe a number of you may have heard of this Florida red tide. If you've ever gone to the Gulf Coast, you may have encountered these things. This time of year, they're actually in full force. And these are large blooms of these little microscopic organisms. And they're small. You can see this cloud of organisms up and down the coast here in the Gulf of Mexico. And you can see the satellite image, and all this red stuff all the way down the Florida Gulf Coast is indicative of these blooms of these algae, specifically something called Karenia brevis. The Karenia brevis produces a particular toxin called brevetoxin, and it's poisonous to humans and to wildlife, particularly fish. So when we have these blooms in the Gulf Coast, the first thing you see are a bunch of dead fish washing up on shore. Okay? But other animals are affected. We have massive deaths of manatees and dolphins that are indirectly exposed to these. Sometimes we lose as much as 10% of the population when these blooms are very bad. Okay? Now, and it also can contaminate shellfish. So it has an effect on things like oysters and particularly in the northern parts of Florida to some extent. These don't have as big an effect as some of these algal toxins do on contamination of shellfish. But they also have effects on human health, particularly if you were to go over there during one of these blooms and they have a hard time breathing. They're very irritating toxins that get mixed up in that ocean spray. And it may not be life-threatening, but it can be at least very convenient, particularly for people with asthma and other compromised pulmonary systems. So this is a big issue, and really that's a big issue for Florida because that means that tourists don't want to go to the beaches. What we do in Florida, we have a lot of tourism. It's our major industry, so the economic impacts of these things can be really quite huge. And I've heard estimates of billions of dollars because of the size of our tourism industry and our reliance on those beaches. So that's kind of a local version of this. Now, as I said though, my favorite group are the cyanobacteria and blue-green algae, and you've probably seen these before too. If you've ever seen green slime in a pond, or a lake, very likely those cyanobacteria, maybe the green algae, but you've probably seen this kind of stuff before. This is actually St. John's River up north here, and they regularly get blooms of these as well, this green stuff floating there. And they're around the world, they're really a global problem. Now they actually exist everywhere in the world, they're really ubiquitous, but they're really most conspicuous when they're associated with these so-called blooms. They can be nuisances, they can be unsightly, and as you'll see when they produce toxins, they can be real health threats. Now it's not just, and these are all freshwater habitats, and they're probably more predominant in freshwater habitats. They're also in marine settings. If any of you are snorkelers, you go out on seagrass beds, you may see these things growing on seagrasses. Some of these are actually dermatotoxins. Just brushing up against them can cause problems. So there can be impact health in lots of different ways. In marine and freshwater habitats, and largely aquatic. But they're not just aquatic organisms. A number of them are involved in symbiotic relationships. If you've ever seen lichen growing in a rock or a tree, it's actually a symbiosis between a fungus and a cyanobacteria. Basically, they've evolved this partnership where the fungus gives it housing, and the cyanobacteria, being photosynthetic, feeds that fungus. Okay? And these things have evolved to almost sort of like super species. And the reality is, the cyanobacteria have been around for a long, long time. So they're really among the oldest organisms on the planet, about 3.5 billion years old. We know that from fossil records, like the one shown here from the Grand Canyon. This is an extinct version of something called stromatolite. It's an extant or a living one, actually in the Bahamas. There's very few of these left worldwide. But these are basically assemblages of cyanobacteria that form these large structures. They used to be very common, and we see the fossil records, again, going back about 3.5 billion years. So really the oldest organisms on the planet. So they've evolved to pretty much every part of this planet. As I always like to say, it's really their world and we're just sort of living in it because we find them everywhere and we see that lots of organisms have sort of adapted to this over time. And I don't have a chance to talk too much about that today. We've got a number of hypotheses to show that really a lot of animals, particularly aquatic animals, have lived to adapt to the fact that they're really living in a milieu of all of these toxic-producing organisms. So, now, we mostly know them from human health effects, or environmental health impacts. As I said, they're most predominant, or at least conspicuous really, in freshwater systems. And that means they have the potential to get into drinking water. And I'll talk a little bit about some of the poisoning cases, and most of them have been associated with contamination of drinking water. If they occur in reservoirs or catchments, and that water is not properly treated, you could be exposed to a number of these toxins. Okay? That's one of the main health concerns. Now, as I said though, just brushing up against them can be a problem. We just advise them to say, don't swim in here when there's green stuff. Because again, just touching these things directly can lead to health impacts as well, health problems. Okay. And this includes aquatic habitat, marine freshwater habitat, as well as marine habitat. And again, I mentioned that one of these seagrasses, Algae, that grow, it can cause sort of dermatotoxic effects. Just brushing up against them can cause irritations from a number of toxins. So these are ways humans are exposed to them. But they also have a big impact on animal health, sometimes commercially relevant animals. So for instance, net pen disease. There was a big die-off of salmon who were exposed during a bloom of cyanobacteria that caused a big impact on the salmon industry. Salmon, as you may know, spend part of their life living up in freshwater habitats. So when they get up there, they're actually farmed in these net pens. And there were blooms of these algae, which led to a big loss of the salmon crop that particular year. But not just sort of domestic animals. Now, I have a picture, I don't think. The other place we see this a lot is dogs get poisoned all the time. We hear about this all the time. You have a dog, and dogs don't care. They're hot. They drink from a pond or a puddle. Steer them away from the green stuff, because we always see this when people show up at vets and their dog is sick. They drank from the pond a couple days ago. It's fast-acting stuff. So even sort of our lovable best friend is getting exposed to these guys, these toxins. It also affects wildlife. Again, an example here in Florida, we had a big die-off of some alligators. It's a place called Lake Griffiths up north. It sort of just shows that a spike in the mortality of these alligators, the belly up alligator, in case it's not clear from the picture. Again, there was a small bloom in that lake, and we had this unprecedented die-off of alligators in that particular body of water. And again, in marine habitats, they are there, and we've had some implications for this for marine animal health. One of these, if you're a scuba diver or snorkeler, you may be aware of something called black band disease. It's really threatening a lot of these coral reefs. This black band disease is the progressive spreading of a microbial assemblage. It's not just cyanobacteria, it's a bunch of microbes that are in here. Teasing it apart is very difficult, but included amongst, you see this black band, this is dead coral, as it's moving its way along here, and included amongst these microbes are toxin-producing cyanobacteria, and there's linkage now that may contribute to this actual pathology of these coral diseases. This is a sea turtle. I mentioned that some of these marine cyanobacteria live in seagrass. The sea turtles love to live in seagrass. And sometimes they encounter these things accidentally as they're feeding on seagrass. It's not really discriminating. The argument has been that some of these same toxins that lead to those dermatotoxic effects can also be tumor promoters and may add to some of those effects. This is a papillomavirus-induced cancer that's found amongst sea turtles. And the idea is that this might exacerbate it by being exposed to tumor-promoting compounds that are found in those cyanobacteria. So far-reaching impacts of these, including human, animal health, both wildlife and domestic animals. Now, by far, though, the reason we're interested in them is because people have gotten very sick from them, particularly by contamination of drinking water. And this is a little timeline going back to about 1878. First guy who kind of pointed this out was a guy by the name of George Francis. And he found one of these little ponds where some livestock were drinking and shaking and keeling over dead. And he wrote a thing called a poison lake. This was in Australia. That was really the first time anybody noticed this. Now, this has occurred, I'm sure, before George Francis came along, but he was the first one to publish his paper in Nature. And since then, every couple of decades or so, we have some of the poisoning incidences, and there's probably more. These are just the ones that kind of make headlines or make it to Nature science journals. So, as you can see, there's a global problem. Back as far as 1931, there was in Charleston, West Virginia, population, a really sizable population there of 60,000. Well, 9,000 people came down with acute gastroenteritis. What happened is there was cyanobacteria in their reservoir. They thought, okay, let's just kill the algae, we'll be fine. So they dumped some copper sulfate out there, which killed the algae, and dumped all the contents, including the toxins, into their drinking water. And they were obviously sorry for that, right? And we see this throughout. I can't go through all of these, but a lot of times it's contaminated drinking water. In one particular interesting case in Brazil, not too long ago, there was actually poorly treated water at a dialysis clinic. Somebody forgot to change out the filter. So you have a rural area where they were dialyzing people. Somebody's job was to go and change the filter so that the water coming in was clean. They didn't do that. About 63 people died because they were basically dialyzed with toxin-laden water. Now, a lot of these toxins are water-soluble toxins, and I'll come back to this point a little later on, but all the ones I show here are kind of what I call the usual suspects. And these are ones that have been associated with some sort of intoxication event. Somebody's gotten sick, died. We know a lot about them. We know a lot about them because they've caused the profound human impacts, acute toxicity. These are just the tip of the iceberg, though. And that's part of the theme of what I want to talk about today, is these are the ones that get on our radar because someone's gotten sick. Part of what we do for toxin discovery is to try to find out all the things we don't know about the world, right? Find out all those biologically active compounds that could be toxic and that might be contributing to these health effects. I also put this up here to summarize, what people have looked at for these. We oftentimes group these things operationally into hepatotoxins and neurotoxic compounds, primarily because those are the two effects that we see. So the biggest one is microsystem LR, or microsystem family in general, are so-called hepatotoxins because they accumulate in the liver and they cause that kind of hepatoenteritis that I mentioned when you drink the water contaminated with these things. Likewise, leucopsin, different molecule, a little alkaloid over here. Likewise, completely chemically different, but also associated with hepatotoxin. Different mechanism of action, but again, because it winds up in your detoxifying organ, in your liver, that's where we see the effects. A couple of these other ones, saxitoxin, for instance, is actually found in shellfish contamination by distantly related organisms, but is a potent inhibitor of neurons. And then this one, anatoxin, also a neurotoxin, originally called the very fast death factor, to give you an idea of how that one works and how it was discovered. Again, I mostly point these out. I'm going to talk more about our new molecules, but most people go into understanding the toxins of cyanobacteria by thinking that this is the world of chemistry of cyanobacteria, and it's far from that. Again, with 3.5 billion years under their belt to make new molecules, they are chemical factories. We can hardly find one that we can't grind up and find something that's toxic or biologically active, and very likely unrelated to this. And that's kind of where we come in now, is to try to discover, explore that chemistry in a little bit more depth. Because the chemistry decides what you see there. Now, I also, as I pointed out, this has been associated with acute toxicity. And it doesn't happen that often. I showed you that through the timeline. You saw every couple of decades. It probably happens more frequently than that. You just don't recognize it. But those are cases where people get sick. They drink water and they go to the doctor the next day. They have gastroenteritis. They have some sort of symptom. We know that there are also these long-term health effects. This is another part of this we don't understand. Apart from the fact that we are just at the tip of the iceberg of chemistry, we don't understand some of these long-term health effects over years and years of being exposed to these things. We're kind of lucky that we have good water treatment here. A lot of people rely on poorly treated water worldwide. It's one of the biggest problems in the world, is access to clean drinking water. And being exposed to low levels of these things, for instance, over time, can lead to things like higher rates of cancer. This is a study that's referenced about in China, where there are folks that sometimes in a year have to use surface water, pond or ditch water, just because of accessibility issues. And they found that those people have about a 24 higher rate of primary liver cancer. This is a hepatotoxin. Where the cancer is happening is in the liver where these things are accumulating. And somebody said, well, maybe that's because they're drinking surface water. Surface water is chock full of stuff. Deep wells, for instance, don't, because these are photosynthetic organisms. They need light. Wells are a bit clean. That's why we prefer wells. Surface water is where you're going to see these algae. And that's where you see that difference in that disease. And sure enough, if you go look at the pond or ditch water versus the wells, there's none of these toxins in the wells. You don't have the cyanobacteria deep down. But you have about 60% of the samples are positive for that microsystem that I mentioned. Okay. And if you kind of do a back-of-the-envelope calculation from the study, it's going to get about 0.19 picograms per day. You guys want picogram, minus, right, this tiny, tiny little bit per day over a part of the year when you have to, maybe during the dry season when you get the surface or ditch water, can lead to these effects. So that small exposure over a long time is the part that's really hard to tease out from all of this. Another example of this, which is kind of coming back into light lately, is the link to neurodegenerative diseases. This is a little story where back in the 50s, when the U.S. took over Guam, took over the protectorate, or what you want to call it, I'm not sure what the classification of it is, they started looking at the locals there. This is Chamorro group, who had this really high rate of something called amyotrophic lateral sclerosis, or Lou Gehrig's disease. And it seemed really bizarre because it's higher than what you normally expect. And they did some genetic studies, they looked at families, there was no connection. And somebody said, this is probably environmental. And they traced it back, it was actually ethnobotanists who traced it back to a cycad first. He said, the cycad was found to produce a compound called BMAA, which is beta-methylalanine. It's basically a weird amino acid. And they knew about it, it's not that potent of a neurotoxic compound, but it is. Turns out it's not produced by the cycad, it's produced by a cyanobacteria called nostoc that lives inside this little cycad, this plant. He said they grind it up and make a flour. So maybe that's it, they're all eating this flour. The levels were not high enough, but it turns out that there's a bat, a fruit bat, that eats the fruits of the cycad, and this fruit bat turns out accumulating thousands of fold more of this toxin. And in Guam, the Chamorros really love this fruit bat. I'm not making that up. I verified this with somebody who lived there. They take the fruit bat, for and all, put it in milk, and it's kind of a delicacy. It's largely hunted out, but that was for a long time a delicacy. And it was suggested that this might be linked to that amyotrophic lateral sclerosis, the presence of that. Now this is sort of the start of this. Since then there's been a study that said that about 95% of all the cyanobacteria might produce this weird amino acid. And now there's studies of people eating reindeer meat eat lichen, and they made this connection that lichen meat reindeer are a source of BMAA. We found this in the brains of people with Alzheimer's and Parkinson's. There's at the University of Miami, they do work with the brain bank there. And they're finding this really closely linked to a lot of these neurodegenerative diseases. And if it pans out to be true, may explain a big percentage of those types of diseases. Again, not real evident, because you don't get this, and the next day you have a neurotoxic effect, right? It takes a long time for this to accumulate. It's only becoming clear now what that mechanism is, potentially is. Okay. And just to round up my sort of introduction to this before I talk about our research, that area of research, this is a problem that's not going away. It's been around for 3.5 billion years, ostensibly. And as I said, they're photosynthetic microorganisms. They just need a little bit of light, like warm water, nitrates and phosphates, the basic building blocks of the plant, and they do well. And we have nitrates and phosphates coming out from agriculture, from sewage, from industrial runoff, from groundwater, and runoff as well. We have warming waters, we have more and more sunlight. We are really fueling these things, so they're a growing problem, and they're becoming more frequent, the occurrence of blooms, and people are becoming more toxic as well. There may be reasons for that. I think this is the last thing. I'm going to talk about toxins. I hate to be the bad news guy. We do molecule discovery as well. And the flip side of this, and I actually will mention one of our examples, is to discover new drugs. The other flip side of this is that some of these compounds may be biologically active in a good way. They may be exploitable as possible drugs. And this is just kind of a smattering of these things that have been looked at as compounds for potential anti-cancer drugs. And there's a number of things that are being looked at preclinically, and I think are starting to enter into clinical trials as possible drugs. So, what we do in my lab, at least in part, is to try to find new molecules, kind of chemical prospecting. And this is just a sort of flow chart of how that works. So we go out, isolate cyanobacteria, we can culture them. They like bacteria, the prokaryotes, we can grow them, little special conditions, not like your usual petri dish, but we can grow them. And then we extract them, take off their chemistry, and screen it. And this is where the biology really comes in. We look for things that are biologically active, toxic or otherwise, and I'll show you the model systems we use. And then we can use that biological activity to ultimately purify the molecule. So we can use that, and I'll show you an example, to move through and find that big pernicious stuff that we extract, maybe 10,000 molecules, to get down to one molecule by using this biological activity as our guide. And ultimately, after that, we characterize it both chemically, we do spectroscopic studies, spectrometry, all those things that we need to do, and also characterize them toxicologically. What are they doing? And I'll give you at least one example of this where we made some headway on understanding how these things are toxic and how they're actually biologically active. Okay. So the first part of this, again, is going out to these field sites, and we in Florida are blessed with a great aquatic habitat for just about anything you want to do. We work a lot in the Everglades. These little parasite mats, who's been to the Everglades? Okay. Still, it's not enough. You live in South Florida, you should spend some time in the Everglades. It's a great resource. And this time of year, if you go out there, the water level's high enough, you'll see these floating mats of gray stuff. And that includes, among other things, cyanobacteria. It's one of the real characteristic communities of the Everglades, in fact. We can go grab handfuls of this stuff. Or we can take water, we can filter if we want to. And we can actually, as I say, culture the stuff on petri dishes and in liquid medium, and we can have tubes and tubes of this stuff growing at any given time, large volumes, small volumes of it. And then we can go start prospecting. And that's where, again, when biology comes in, screening for biological activity, and again, using that to get to a pure molecule that we can characterize. Okay. And this just sort of shows how well we do this. We take a lot of green stuff, our agriculture, we call it biomass, that's the cells and all the stuff that's there. And we grind it up into non-polar solvents or polar solvents, in this case chlorine or methanol that we typically use. And that's going to give us a good representation of all the chemistry that's there. And a lot of what we talk about really relies on this part. Okay, so if you take chemistry, you probably encounter things extracted water. For instance, you have a tea, you're making water extract, right? Those are more polar molecules. If you were to extract the same thing in oil, you get off a whole bunch of different molecules, right? And that's the so-called lipophilic fraction. And I'll come back to this because that's really our area of interest right now, one of our major areas of interest. Well, we can get both of these extracts again in our screening process. We don't know what we're looking for, so we scan out everything, the best representation of everything, and try to look for some biological activity. This is some old data, and I'll use it more as an example, where we screen a bunch of things. And you see the one in red there shows this good antimicrobial cytotoxicity across the board, lit up, very potent activity. That goes to the top of our list. Once we have identified something like this, we do that bioassay guided fractionation. Don't worry about the details. Basically, it took us a year and a half or so of my time. At that point, I was still working in the lab. And we went through all this stuff, so you kind of bang your head against the wall, figure out the best path to get there. But you use that biological activity, follow through all those steps, until you ultimately wind up with something that gives you a pure compound. And then you characterize it. Now, in this case, we were using cancer cells. This is an assay showing these are cancer cells going in a dish when they're blue, they're dead, whether pink, they're alive. So we can actually then test the biological activity of these along the way, and it led us to this compound, something called the pahyopolides. There's actually two of them, two variants. They're called the pahyopolides because they're isolated from the Everglades. The biggest name for the Everglades is the pahyopi. Because it's such a big structure, if you give the actual chemical name for it, it would be 3 pages long probably. We have to come up with these trivial names. We name them after where they're isolated. In this case, pahyopi and the pahyopolides were born. I said it took about a year and a half to purify it. It took us about 3-4 years to get that structure. It's not easy. And people smarter than I am were involved in this. This is a group effort. But it was a new molecule, interesting. It turns out that it has differential activity against a bunch of cancer cells, cancer types. That's what you want to see with an anti-cancer drug. You don't want to kill all cells the same. You want to specify certain types of cells. So it showed that kind of differential activity and promise. Last I heard, there's a bunch of group that are trying to kind of tease this apart and tweak it to make a potential drug for an anti-cancer drug. We kind of left behind that because I kind of moved on now to what I'll talk about primarily now and forward is this zebrafish embryo model. And because we're interested in things that are environmentally relevant, cancer cells are not environmentally relevant, right? They're relevant to human health. That's great. We can always come back to that. But we wanted something that would really approach environmental toxins. And because of that, and because of a number of practical advantages, we work primarily in this zebrafish embryo system. Now, you've probably all seen zebrafish before. Every Petco has them, I think. You put them in there like that, you put them in your aquarium, they're feeder fish. In fact, we get some of these from Petco. They work out pretty well. The reason we like them is because their embryos and their eggs are really easy to work with for a number of reasons. First of all, they're small enough, but they're not too small. They're about 1 millimeter in diameter, which means that you can put a bunch of them in little wells and test a bunch of them at one time. They're not so small that you can't see them. They're easy to raise. These things are not hard to fish to raise. That's why they have them in Petco's. Most importantly, they have a nearly transparent embryo. So we can see, and I'll show you a video here in a second, once they're fertilized from a fertilized egg to a little baby fish, we can watch that whole thing happen. Okay? And there's a whole bunch of stuff happening there. Right? So apart from these other things, you get a lot of eggs, the genome's been sequenced. I like to emphasize this one, that it's really a high output model, because what we're looking at is a dynamic process of who knows how many biological processes that are occurring, all of which might be potential toxin targets or drug targets for that matter. It's also a drug model, for new drugs. So, as I said, this is just an orient tube. This is a 2-cell that came from a fertilized egg. So you first have a fertilized egg, and then cell divides to 2, becomes 4, 8, 16, that from biology, right? That's the fish out. Then you get a big ball of cells, and that big ball of cells starts to turn into an organism. This happens in the case of the zebrafish in about 3 to 5 days. Now, it's not fully grown, but you get a baby fish, and all the little parts are there. And I'll show you that. So these are those first 2 cells. This baking, by the way, is just a yolk sac. So early on, it has to feed off these nutrients from its mother, right? So it's that little care package that it has for those first 3 to 5 days to feed off of. So what will happen over that, this is about the first 24 hours, is that 2 cells become 4 cells, 8 cells, 16 cells. And over time, they'll start to wind up with thousands of cells. This big ball of cells still feeding off that yolk sac. And then at some point, something turns on and tells this ball of cells, start differentiating into something that is like an organism, that has a body axis, that has an anterior and a posterior end, and start differentiating into organs like eyes and some of these somas that are important in the nervous system. And all this happens just the first 24 hours. And this time lapse, obviously. But if we wanted to, we can sit there under the microscope and watch all of that stuff occurring and look for any problems in that as possible impacts from our toxin exposures. And we look for things. This is an example. It's actually not mine, but here you see the so-called somites in one versus the other. We look for differences between our controls and our embryos that are treated with extracts or pure compounds. Now, this is some data that we had, some pictures from some of these things. We started screening these things. We came up with we have probably about 20% of them we looked at gave us some biological activity in the system. Interestingly, they affect early stages, mid stages, and late stages of this. And these would be that first 12 hours or so. Some of them never got past that ball of cell. They get stuck there, which is actually interesting because that's where they start making their own RNA. And it tells you a little bit about maybe what's happening, why the toxin, what the toxin's doing. Some of them get past that, but they never differentiate their head or any of those organs. There's weird effects, and we see this quite a bit, and I'll come back at the end. I think it's because there's some widespread chemistry there that gives this sort of what's called phenotype, or we call it toxotype. It's not really genetically derived. It's derived from the toxin exposure. And then later on, some of these things make it past that. And these are living fish. They just have these edemas and these deformities, all of them. So they're bent bodies, big swollen parts of them. So we can see all these things being affected. We really get excited when we see things that are consistent. So these are normal embryos. So our control at 24 hours will look something like this. Here's your eye, your body acts, tail wrapped around the yolk. This is normal zebrafish is about 5 days maybe, swimming around. At some point we'll start eating when it's available. And these will be just several normal ones, but at that same time, here's ones that are all deformed, but they're all deformed in the same way, which tells us that there's a particular pathway that's being affected. We don't know what that is, and that's the hard part later on, but we have something that we know can be associated with a toxicity, a biological process. And we can use that then to kind of purify these compounds and characterize them. One that we first came up with was this Fisherella. This is a filamentous cyanobacteria, fairly widespread. It's its own kind of weird little order within the cyanobacteria. And when we exposed the embryos to these extracts, these would be normal, say 24-hour embryos. These are the ones 24 hours exposed to the extract. Everything's forming more or less normal, but they lack this pigmentation. The cells are a little funny. It depends on the dose and that kind of thing, but there's these very characteristic ghost-like toxotypes that we see because of exposures. And you could do 1,000 of them, they would all look like that. And you start to speculate on maybe what the mechanism is for that toxicity. But we can use this toxotype, this manifestation of this toxicity, to guide purification. The student, Katie, who worked on this, did this, and she did one of these cultures, and she looked at that globuline, some of the biomass, and the liquid that's floating around in the culture medium. What she interestingly found is that there's this extracellular stuff that kind of oozes out of the cell and sticks to the flask. It was rich with these toxins. This activity was mostly in that goo that came out of those cells. It has also made purification much simpler. As opposed to that big flow chart I showed you, she was able to do a couple of steps and purify this stuff. And we always like to go first for the low-hanging fruit, you know. Once we get a handle on it, then we can work backwards and look at the more difficult stuff. So she purified a number of compounds. The two major ones were these two guys here. But once we have them purified, we can go back into our biological system. And it turns out the one on the right, in this case, was really the one that explained this best. Okay? There were some effects of this one, but not as much. So we're going to be tied to a certain compound, and we can start to speculate when we have this in hand, you know, what are the chemical differences between these guys. We don't need to be a chemist to understand they're chemically different. And when we see that there's a basis for this similarity, there's a lot of similarity to those two molecules, but modifying a group here or there can make these things quite different in terms of how toxic they are, or how they are toxic. The interesting thing, little side note, as I move on, is that we also came across the same strain when we were looking for things that inhibited other algae. One of the reasons, we don't know why a lot of these compounds are produced, to be honest with you. But one suggestion has been that they are produced to inhibit other algae in the environment, to give them a competitive edge. You know, it's the idea of so-called allelochemistry, or allelopathy is the term that we use. So we were doing experiments to do this. We tested all these Everglades isolates against each other. Basically, what we saw is Fisherella was really the one that inhibited all the algae across the board. Okay. And in fact, these compounds had been previously isolated as antimicrobial compounds. And the argument goes that they're producing them for some functional role to inhibit other algae, but when humans or zebrafish might be exposed to them, there's toxicity to those vertebrates. So it's a coincidental toxicity. Now, for the last bit here, I'm going to get to this concept I talked about, which is this lipophilic chemistry. And I put these usual suspects up there. They're all water-soluble compounds. They've all been associated with contamination of drinking water. Well-studied, most of them. But these are not the whole story, okay? And we know this, other people know this. If you take a pure toxin and you test whatever biological activity, and you take a crude extract from wherever that organism, that toxin came from, usually that extract is more toxic, or has added toxicity, or added effects to it, which implies that there's other stuff in that mixture of compounds. And people kind of usually ignore them because they know about these 4 or 5 or 6 things, and they always want to try to link this, you know, easily. So we have put there a zebrafish model. So I'm looking at this compound solution for MOPS, a water-soluble alkaloid, certainly toxic, inhibits protein synthesis, and we put it on our zebrafish. First, didn't do anything. Turns out that this water-soluble thing doesn't like to cross with the membranes of our zebrafish, which is a limitation, maybe, but in our case it was serendipitous because you know, we can make this toxic. We can take this stuff and actually get a little tiny needle and inject each one of those little embryos, and we can get these kill curves. And sure enough, it kills the embryos at a certain dosage. That's all it's showing you. Amount required to kill a certain number of embryos would be lethal to 50% of them, or something called LC50. We can make it work, but if you put them, if you bathe them in silica frost at high concentration, it doesn't get in, it doesn't cause a problem. Now, if we take an extract from one of the organisms that produce this, and we bathe them in that extract, we start to see toxicity. So these are number strains. Only 2 of these produce silica frost. All the extracts that we put them in gave us pronounced developmental toxicity in our zebrafish embryo, suggesting that this is not a developmental toxin. There's something else in there. And it turns out that's really associated again with that lipophilic fraction. Okay? And we see that there's oftentimes this lipophilic chemistry that really has been overlooked in the past decades. Let's show examples of that toxicity. So this was first for a couple strains here. We went through these things in lipophilic extracts, and sure enough, the lipophilic extracts were active. We went back to some of these known toxin-producing type species, ones that are associated with toxicity, but are associated with maybe this water-soluble toxin or this water-soluble toxin or some of those things. When we started looking at the lipophilic chemistry, which would not include those toxins, they were all producing something that was biologically active and really hadn't been looked at before. Okay? It's not in the right way. This is where our zebrafish embryo is great, because lipophilic stuff loves to get into it. So we have kind of a way that we now can really sift out all that water-soluble stuff and really look at those lipophilic compounds. So Asha, as I pointed out at the beginning, has been doing this. This just shows the biological activity, normal embryos. And she used this biological activity, toxicity. Sometimes it's very general, sometimes it's specific. She's seen effects, particularly in heart development, which kind of was pointed out here, which is interesting enough. But again, the low-lying fruit, the low-hanging fruit, we looked at this one called phenazonamide. Fairly simple to purify. What she pulled out was a family of compounds called polymethoxy alkenes. They're shown here because they're polymethoxylase in the chemistries of the methoxy group. And depending on how long this is, there's about 6 of these for this particular species. And it's interesting. Okay, it's potentially a new family of cyanobacterial toxins. But so then we started looking at, these are all the ones that were found from that particular species. Here's all the different variants we started looking for. And we started looking at some of those other toxigenic species, like the Silligeromopsis raceborski, usually associated with Silligeromopsis. Sure enough, it had some of those. Microcystis produces some of those. They seem to be widespread amongst those groups that I told you before. Moreover, when we start going back to the literature, people had purified them before. They just never knew they were toxic. They accidentally purified these. Going back to 1979, people have shown these very distantly related groups of cyanobacteria, suggesting these are a widespread component of the toxicity of the cyanobacteria in general. Okay. And this is a quick slide showing that we also know that these wind up in other organisms. The only other place they've been isolated is the sponge, and almost definitely this sponge was eating cyanobacteria and picked up that toxin. So they're there, they're present, and they're available in the environment. We wouldn't have known that if we didn't start looking with the right set of eyes. And it's the last one we'll talk about, because again, a lot of times we look at these things as they must be components that are what we call secondary metabolites, things that are just there specifically for some function in order to be toxic. So when Asha started doing this stuff, she kept pulling out this compound. It was a red-colored compound. And we always use color to kind of separate stuff when we do chromatography, and I would say, the red stuff is probably some pigment. It doesn't matter, it's just a photosynthetic pigment. It can't be that compound. She just kept seeing the red stuff. Ultimately, she couldn't deny it anymore. She pulled out this compound called mixoxanthophyll. Now, mixoxanthophyll belongs to a group of compounds called the carotenoids. This is, again, it was toxic in our embryo. I'll show you pictures of this. These carotenoids are found all over. They're found in plants. They're pretty much found in all photosynthetic organisms. And they're accessory pigments. They help chlorophyll out. They collect light. And I think I have a picture in here. There's a hot springs, famous hot springs out west. And this is some brilliant colors, all from those pigments from cyanobacteria. They're key parts of the pigmentation of these photosynthetic organisms. They're involved in so-called photooxidative stress. They're there so when light's pounding on you and causing oxidative molecules. It protects against those. It's an important part of photosynthesis. And just general pigmentation, widespread. And we always thought the only thing that they could possibly be doing is making these things colored, protecting them against light and oxidative stress. How can they be biologically active? Well, and here I'm going to kind of use some speculation. This is our working hypothesis, and this is new stuff. So hopefully next year you'll read about this in Nature. That's my goal, right? So you get the first appearance, right? This is on tape, so it doesn't get out. These xanthophylls, like mixoxanthophylls, belong to this family called the carotenoids. These carotenoids have this long chain of what we call conjugated double bonds. And beta-carotene is the one probably that's in carrots. This is the stuff that's it's so-called provitamin A. Because what happens in beta-carotene is this provitamin A, there's something called monooxygenase. You put a little oxygen here, and then it leads to a possible place where this can break. And what happens is this thing cleaves into something called retinol. So that's just about half of the molecule that's left behind. Now, retinol can get turned into retinol, which is the substance in your eyes, and basically it's vitamin A. So vitamin A can be derived from your diet by this process. Now, there's another step, another oxygenation that can occur, and we know about this, and It's kind of a dirty little secret of the chemistry world. People form this thing called retinoic acid. Now, retinoic acid is important too. This is the stuff that winds up in your eyes, responsible for good sight, and all of a sudden you eat your carrots, you're going to have better vision because you're getting vitamin A, right? Retinoic acid is also an important molecule. We need this as well, particularly during development. It's one of its key signaling molecules during embryo development. And embryos produce it, zebrafish produce it to some extent in their own, because it coordinates, in particular, the development of that body axis. Remember, the body axis sort of spread out and get anterior and posterior? That's actually in part regulated by this thing called retinoic acid. Okay? And the idea is that retinoic acid binds to a particular enzyme, or to the protein, a transcription factor called retinoic acid receptor. This interacts with this is all fairly well described previously. It interacts with a transcription factor that can bind to certain genes and essentially turn those genes on. It's the cue that turns those genes on. And it's particularly a set of genes called the homeobox genes, or the Hox genes. Which again, it's separately been very well described because those are the things that lay out that body plan of an organism when it's developing. You know, from fruit flies to zebrafish to human eyes. Very important set of genes for development. And sure, not being any problem with transcription factors. These are mutants of zebrafish. If they have a problem with these transcription factors that bind retinoic acid, they don't differentiate well. They get these short little bodies, and they just don't elongate. And lo and behold, we say, well, what do ours look like? Do we match up with this, or can we explain it? Because our thinking is that this is the carotenoid. It's not the same. It's not beta-carotene. But monoxidase, we know, can interact with these enzymes. They can cleave them, and they can produce some derivatives of these things, maybe hydroxylated versions of those retinoids, we call it, or maybe new retinoids, we don't know. But we know that chemistry a lot of times, if you have something similar, may act better, it may be an inhibitor of a known process. And so our working hypothesis was that, hey, maybe these things actually create some retinoid that causes that thing to not develop. It inhibits that Hox gene, it inhibits those genes that are responsible. People have shown if you dump a bunch of retinoic acid on them, on the embryos, they get short, like if they had a problem with those homeobox genes. Ours, that's what these are. This is retinoic acid exposed embryos. That's our exposed embryo. And we see that a lot. That same lack of differentiation of body plan, that short tail, just like we see if we were to dump on this retinoic acid at high concentrations. Suggesting that we might have a pathway here. Not only do we have a pathway, because these carotenoids are so widespread, they're probably very widespread in the environment. And again, something that was there the whole time, for 3.5 billion years, right? And explain toxicity people just never thought about before. Just to round this out, there was a lake in China, Lake Tide, which is plagued by this bloom of cyanobacteria. And one of the things they keep finding in this bloom are little amphibians that all come out short, poorly developed, like this, because they're being steeped in this toxin. And people are kind of scratching their head why that might happen. But that's probably because they're in this toxic milieu. So, I don't want to go too long. I'm done here for this part. I just want to conclude. My conclusions are really kind of general. These things are a rich source of biologically active compounds. And our model, we think, is a very good model for probing this for lots of different reasons. And that really more than anything, there's a huge diversity of chemistry that's out there still being explored with regards to this group of cyanobacteria, this group of organisms. Okay? All right. Open for questions. Anyone have a question for the speaker?
So the beta-carotene, are you talking about is that for the fish or is that for humans?
Well, in humans, so all animals need retinol, which is vitamin A, right? And one of the ways you get that is through your diet. And one of the ways it comes from are these pigments that are from plants. So for instance, carrots are chock full of beta-carotene and other things as well that can be used for this. It's a dietary source of other animals. The thing is, some of these byproducts, like the retinoic acid, which is not vitamin A, it's closely related, although they're also important for humans and fish and else. They're sort of a developmental cue, a chemical cue that coordinates all that embryo development. If you get overdosed with those things, we've seen that, we don't do this with humans, of course, but you probably do the same thing if you were looking at a developing human, have this lack of proper development. And what we're saying is that we're kind of giving them a dose of something that normally they would get a high dose of. So we're giving a new source. It's not that compound, that xanthophyll, but rather the fish is turning it into something. There's actually an enzyme in the fish, or in animals, we all have it, called this monooxygenase that converts that beta-carotene into, in the case of beta-carotene, into vitamin A. In the case of other things, related compounds, which may actually be, as we're arguing here, toxic variants on that vitamin A. That can answer your question?
One of your early slides had satellite data.
Yeah.
How do the satellites measure?
So they look at, yeah, oh my God, take me a while, let me do it this way. Yeah, so you can actually look at chlorophyll. So one of the things you can do is you can look at light, I guess not my area, but the general gist of this whole thing is that chlorophyll is a pigment, right? And that pigment has a particular absorbance to it. And satellites can just differentiate that. And all of this basically is coming from chlorophyll, which in this case, we know now matches up with the spread of those actual blooms. You go on the ground and you actually verify this. But what you're actually doing is, yeah, looking at that light absorbance of chlorophyll, which in this case would be a high dominance of photosynthetic algae that are coming down the coast. And in this case, the red tide algae, the brevis. So the Greek magics of satellite technology, NASA's got a program that should do this kind of stuff. To go watch these things from above. Answer everything.
Any other questions?
Oh,
One more question for you. You mentioned earlier that certain mammals like dogs, et cetera, can have toxic effects from consuming cyanobacteria lakes, streams.
right. Contaminated water, yeah.
Of course. What are some symptoms of that?
Well, again, these are hepatotoxic effects. Usually the thing is liver failure. In the case when I've talked to vets who encounter this, because they wind up in the liver, it's not because the liver is more sensitive, it's just that when your liver does, it tries to pump everything out. That's a detoxifying organ. And there's actually organic transporter proteins, they call them, they're pumping through the hepatocyte, unknowingly pumping them full of toxins. So usually, yeah, usually liver failure is one of the key things, and it's obviously kind of malaise, and they don't eat, and they kind of, yeah. If your liver stops working, it's a problem.
Don't feel so good.
Yeah, you don't feel so good.
Oh, yeah.
Leads to other things, yeah.
Can you describe the process of how you actually found the
Well, once we get a pure molecule, that's that scheme I showed you is just using biological activity, taking 10,000 molecules, separating into 10 pots of 1,000, stay those 1,000, get it into 10 pots of 100. That's kind of what we're doing when we purify stuff by, I guess, some column chromatography, maybe, if you're thinking, well, if not, basically we separate by all sorts of different methods and try to distinguish all those compounds until we get to the point that we have one. And we,
that's the one.
because we use that biological activity. So let's say we take 10,000 molecules, we somehow separate them into 10 groups of powder, but something like that. We tested one of those 10 groups. This 1,000 are the ones that are biologically active. And then we separate those into 10 groups of 100, tested one of those 10 groups. This is the one we picked it to. It's not always that easy. You can get a little greasy and a little weird in there. That's the part that's hard. Because you don't know what you're looking for, right? You're just using your eyes are the biological activity. Your eyes are the toxicity. And each time you separate that into smaller and smaller pots of things, you test it and test for that particular biological activity until you look. And there's ways you can look at it by seeing that there's just one thing left in there. That's got to be the compound. Then it gets fun. Then the chemistry starts in. That's why I say that part's not trivial. NMR spectroscopy, IR spectroscopy, UV, all of these things, we have very instrumental techniques where we look at the various features of that say something about the structure of that molecule. That's give you a whole course on that. Yeah, that's the general thing. Good, that was questions. All right.
For the red tide, why is it that it's only periodically? Like, why does it come? Why is it always there?
Good question. So if you folks who are studying this, and as I said, photosynthetics, they rely a lot on these nutrients. And we know that they happen around the end of summer. In Florida, that's probably because what happens in the summer here? It's hot and rain. It rains like crazy here, right? And what happens is all that stuff winds up running off in the ocean. And that carries all that agricultural stuff, all that stuff. We see big spikes around times when there's hurricanes. We really get a lot of rain. That's when we see some really profound red tide. So the current thinking is that it's pushing all the nutrients out of the Gulf. They're already there. They're always there all year round. Little levels of them floating around. But when you get all the nutrients out there, as the theory goes, then they're happening. Lots of food, they start to multiply. They can actually live in the silt for a long, long time, sleeping until conditions are right, and then they all come up and they form those massive blooms in the ship. So that's the general thinking. Yeah.
I just wanted to know for the retinol acid, because you said that it shortens the embryo.
Yeah, so you remember that video where they kind of start to go like this?
Yeah.
That's actually coordinated by a set of genes that tell you to kind of sort of differentiate. And fruit flies are another model they look at. Fruit flies are little insects. They actually have segments in them. And those segments are distinguished by a set of genes that all tell them this segment, this segment. We don't have segments visible in fish, but that's kind of the same concept, is it lays out that body plan. And our theory is that what's happening is that because of the chemistry is very similar, that what it's doing is preventing those genes from telling the embryo to start to elongate, make a head and a tail, and all the parts that go in between. And that's a fundamental part of dividing.
Are they being linked to the, like, I guess, the misdevelopment in human?
Yeah, and it could. So that's the thing. There's evidence to suggest that people who are exposed, live near freshwater bodies with blooms. A lot of things have been done in Australia with this. They've seen lower birth weight. They've seen some of these effects in populations that are living close to water, Aboriginal populations in particular, that have been studied with. And so they've seen all these sort of indicators of birth defects or various problems because they're exposed. They don't really know why. There's no real connection between toxin X and that happening. We think this may be one of those things that can contribute to it. Because if they are so widespread, you don't have to be some exotic cyanobacteria in order to be toxic. You can just be any old cyanobacteria and potentially have that toxic potential.
Well, you kind of answered one of them already, but the other question was, recently, well, not that long ago, the news shows about a lot of fish that were dying along with sea and a lot of birds. Could that be one of the causes?
Well, it depends what the fish were.
Like a little central.
Was it here in Florida? Was it on a lake or on the marine habitat?
Central United States.
The central, okay. Well, because there are some. There's a case called the Mulligas. This is not cyanobacterial toxin, another algal toxin. You ever see the movie The Birds? The Hitchcock movie? Apparently, this came from the Mulligas because fish will accumulate this toxin, and pelicans will eat fish, right? And apparently Alfred Hitchcock, as the story goes, read about this in the newspaper, that there were these pelicans walking around all disoriented. And apparently that was his impetus to write The Birds and come up with that story of the birds. So that could have been one, but this happens a lot. For instance, in freshwater systems, there's certainly aquatic birds, ducks. I have a great picture of ducks swimming around and eating stuff. So that certainly could be one of those cases. And with fish, for sure, there's been lots of fish die-offs. Here in Florida, St. John's River up north, for instance, like belly up fish because they're swimming in green, floating in green stuff. Yeah. So that's where we come in. Okay, what's here is try to pull apart. It's not always easy to find out what the thing is, or what the 10 things are that are causing that. Great.
Like an increasing incident now, or?
That's the idea, is all the conditions are right, and that we see them more and more frequently. For instance, we work a little bit in the Great Lakes. They kind of disappeared for a while in the Great Lakes, and now we're seeing them every year become back again. We see blooms in the summer when water is warm, lots of runoff, lots of sewage in particular up there in Lake Erie. And that's the idea, is that we're giving them all the things they need. Global climate change is giving them more water. Lack of, whatever, sunlight, they say, the ozone layer is giving more sunlight. And all of those nitrates and phosphates are part of agricultural fertilizer, they're part of sewage, they're part of all sorts of things that we put into the water.
So they're not just growing here, they're growing all around the world.
Yeah, oh yeah, that's not Florida, that's not the US, and that's absolutely global. Yeah.
The cyanobacteria water, could you get rid of it by avoiding?
Good question. Probably, but it'll be good. You probably get some fraction. A lot of these are fairly stable. Some of them are not. Some of them are quite unstable, including neurotoxic compounds in particular. But there's been a study. I say that because these also get into fish. There's some studies that suggest they get into seafood and fish as well. And at least one study I saw that made the fish more toxic when they cooked the fish. And the reason is that in this case, a lot of the toxin winds up being buried in the fish. But when you sort of cook it, you break up all the proteins that are stuck. It's kind of the idea. And you release the toxin. But the toxin isn't affected at all. It just got released by cooking or anything. They did like boiled carp or something. They did this experimental study. So they're fairly temperature stable. I mean, if you boil it long enough, you might. But yeah. Now again, water is generally treated, and there's like limitations here. But I wouldn't worry too much about it in your case. Although we had some levels of this in Orlando not a few years back, they reported levels above the World Health Organization guidelines. And nobody got sick as far as I know, but it does happen, at least in the reservoirs that the water comes from. And hopefully the water treatment gets rid of it. Gets rid of what we know. Yep.
Can cyanobacteria grow the same way in a fish tank, for example, inside a house, as opposed to outside?
Can you get these in your fish tank at home?
No, but,
Yeah, you can, that does happen, yeah. Yeah, I mean, certainly, we grow them, they're pretty easy to grow. Some of them you can't. Now, all microbes are tricky. We can only get about 1% of all the microbes in the world can be grown. That's a biological reality. But the ones that we grow, room temperature's fine. Light that can be too intense, we put a molten cloth over them, or adjust the lights in our chambers. Give them this BG11 medium and they're pretty happy. So you can probably grow them in a fish tank. And I think you can get some of these would potentially, if you got them in your fish tank, for instance, you know, they could be a problem for your fish tank, yeah. I mean, I guess if you took outside water or something weird like that, I don't know how to get it out in the first place, but it certainly could happen.
it was just because you said that it needs sunlight mainly, and you know, sometimes with
Yeah.
fish tank, but.
Well, we grow all behind laboratory light, ambient light. This is probably perfect for them right here. The medicinal property, sure. This is the one that we kind of pulled out as an anti-cancer. A lot of the drive to biologically active compounds for drugs has been driven by anti-cancer drugs. And I'll tell you why, because it's kind of easy to look for. You can grow cancer cells. We do this. You put stuff on there, you can find things that kill cancer cells. It gives you a lead, right? If you have a very specific disease, like diabetes or something, it's not just, you know, cancer is not curable so far, right? But it's a relatively simple concept. Cells grow, grow, and grow. So finding something that stops cells from growing is a big target to work for. When you look at something that's much more poorly understood, it's hard to get to those diseases. That's why. So that's one thing I would say. A lot of this is driven by that. So the ones I put up there, when we looked at, we did that because it's easy. We find things that I can't say that's going to be the next cancer drug because there's got a lot of stuff you got to do after that, right? I teach a course on this actually, and it's going from the discovery to preclinical. It costs the dollars. It takes decades to get to that point because you've got to do all the studies, but it's a good place to start. So a lot of the stuff starts here by finding biologically active. I keep using the term biologically active to qualify that. It just means something that has an effect on a biological process. So a lot of the medicinal stuff starts there. Now, as far as medicinal people using these, they have spirulina. These health nuts out there. Spirulina is one of these things you can buy at drugstore still. Basically, there's a lake called Lake Klamath in Oregon, and they harvest the stuff and they put it in pills. It's supposed to have all these health benefits. They don't know why it is. I'm not sure there's anything to it, but you go into any Walgreens and look at dietary supplements, you'll find spirulina this, spirulina that. I always found spirulina pasta at a place out in California. Pasta, it's really bizarre. So yeah, there's not a lot of, like in plants on the other hand, a lot of it's been driven by ethnobotany, so you grind up this plant and make a tea. We don't really have many examples of that.
All right, if you guys would, well, join me in
talk generally, casually about research and think about ahead.
Yeah, we're just going to change rooms. If you want to join us, hang out here. You can follow us over. We can meet us over.